So the incubation time for fungi is generally 3 to 5 days. Some fungi they take several weeks to grow and to form visible colonies on the agar medium, and generally yeast it will take 2 to 3 days for the colonies to form on the Sebarodextrose or potato dextrose agar medium. So this is about the Sebarod's dextrose agar medium, which is used to cultivate fungi, yeast, and several different kinds of molds. So this is an image which is representing the Sebarod's dextrose agar and yeast growing on it towards left side. So this one is a a Severot's dextrose agar media on which yeast is growing. So you can see the difference, observe the difference here for Candida albicans and one of the other fungi, that is Trichophyton terrestre, which is growing on Severot dextrose agar media. So you can see the difference here. So this is how you identify yeast. You can see yeast here. So it looks something like bacterial culture or bacterial colony. So but it is not bacterial colony. Okay, so it is a yeast which is growing on a fungal medium, STA, that is STA. They are generally creamy in color and they are round and very creamy or white to cream in color, and they usually take 2 to 3 days for growth to appear on this STA media when it has been cultivated. So this on the towards the right side you can see Candida albicans and the other one is Trichophyton terrestre. So this is how for the visible colonies to be formed on this Severodextrose agar media, it takes in order to be visible. So it will take several days, that is around 7 days, 1 week to or more than 1 week. It will take depending upon the availability of the nutrients and the kind of or the type of fungi which has been growing on that particular media. So this is about the images, that is the yeast, and this is the other examples of fungi which is growing on Sebarodextrose agar media. So we can grow this, cultivate this kind of fungi in our laboratory. So the next media is potato dextrose agar media. So it is also one of the basic medium which is used for cultivation of fungi, yeast, and other different kinds of molds. So PDA stands for potato dextrose agar media. The source of carbohydrate or carbon source is dextrose, like in Severod dextrose agar media, and it is serving as a growth stimulant. And we also have potato, that is the infusion of potato is used here. That is the procedure of infusion is nothing but we can prepare it. So if you are getting a ready-made potato dextrose agar media, so you get in the form of powder, but we also can prepare potato infusion, that is potato dextrose agar can be prepared in our laboratory. The infusion or the preparation process of potato is you can make it into slices. The potato can be made into slices. Take some amount of water, put the slices in the water and boil it for some time, and then you can take a muslin cloth or cheese cloth and you can filter it. So the filtered whatever is obtained, that is being used, not the pieces of the potato or the slices of the potato, but that is infusion. Okay, so what is the purpose of this potato infusion? It provides a nutrient base. That means by adding this potato in this media, so it is acting as a rich base. That is, the potato infusion is acting as a nutritional rich base which will encourage the mold sporulation and even pigment production. So some of the dermophytes, they are also able to produce pigments, and that is how you can identify what kind of fungi it is. Okay, so like how bacteria are able to produce pigments when we grow on different kind of media, even fungi are also able to produce pigments, and that is how you will be able to identify it. So potato is acting as a nutritional source for the growth of yeast, fungi, and other molds. So generally agar is added as a solidifying agent. And next, coming to the composition of potato dextrose agar, is we have potato infusion, dextrose, and distilled water, and the pH of this PDA is similar to that of SDA, that is Severod's dextrose agar, which is 5.6, because fungi, yeast, they prefer to grow at acidic pH, and we are adding solidifying agent or gelling agent, that is agar powder. The procedure for preparation of this PDA is similar to that of other kind of media, but the only thing is here we are trying to prepare potato infusion is by boiling some amount or some quantity of the sliced or unpeeled, or you can also take peeled off, because we are putting it for sterilization. You can also use unpeeled, otherwise peeled potato slices can also be used. So because what happens is when you do not use the unpeeled when you use the unpeeled potatoes, what happens is usually soil or mud, whatever is present on the peel, it is also acting as a source of microorganisms. But as you keep it for what do you say, sterilization, and during sterilization process, whatever the soil containing microorganisms on the peel, on the cover of the potato, can be killed off or inhibited. So, and this is how we are taking some 200 grams of sliced potato slices, boiling them in 1 liter distilled water for 30 minutes, and then we are filtering with a cheesecloth and saving the effluent, which is nothing but the potato infusion. And then we are mixing dextrose, agar, and water. Remaining water, whatever is there, we are adding, and then we are autoclaving it, and then dispense into the petri plates and can use for cultivation of the fungi. Now, next, coming to selective medium and differential medium. So here, that is McConkey's agar. So McConkey's agar is acting as both selective medium and a differential medium. So what is the specialty of this McConkey's agar medium? This is especially used to grow Enterobacteriaceae members, that is intestinal bacteria, especially intestinal pathogens and coliform bacteria. This media is generally used to isolate intestinal pathogens or coliforms. It is acting as both. So if you are asked about which media is acting as both selective and differential media, your answer must be McConkey's agar. Why and how it is acting as a selective medium and differential medium? So for Enterobacteriaceae members, it is acting as a selective medium. For lactose fermenters and non-lactose fermenters, it is acting as a differential medium. So here, now if you see, so neutral red is an indicator, that is a pH indicator, which will turn red at pH below 6 or below 6. Usually at this pH, the neutral red is going to turn red, and is usually colorless at any pH greater than 6. So this is a specialty of neutral red. So if we see the composition, we have neutral red here. Neutral red is also been added into this medium. It is acting as an indicator. So because of the addition or presence of this indicator, it is able to differentiate lactose fermenters and non-lactose fermenters. So those which ferment lactose, we also have lactose as a source of carbohydrate. So those organisms which are able to ferment lactose, that is if they are able to utilize the lactose and ferment it, then as a result of fermentation, some end products are formed, that is acid. So here when neutral red is added here, this neutral red, what it is doing at lower pH or pH below 6, what it is doing is it is turning into red. But those which are non-fermenters, that means those microorganisms or bacteria which are not able to ferment the lactose, they do not turn red, they remain colorless, that is white or cream in color. Why? Because they are not fermenting or they are not utilizing the lactose carbohydrate, neither any end products like acids are produced. So because there is no production of acids by that particular bacteria, that is non-lactose fermenters, the color of the colony will be white or cream in color. So this is how it is acting as a differential media, that is McConkey's agar. It is able to differentiate between lactose fermenters and non-lactose fermenters. Now next, coming to the other one, that is bile salts. The presence of the bile salts will make the medium selective for Enterobacteriaceae members. That means because of the presence of the bile salts, and crystal violet is also been added, that will inhibit most of the gram positive bacteria, and which will allow only gram negative bacteria, because coliforms are generally gram negative organisms. So the presence of this bile salts, that is sodium taurocholate, it is making the medium selective only for gram negative bacteria and inhibiting gram positive bacteria. So it is generally used to isolate members of the family Enterobacteriaceae. So coming to the components or composition of McConkey's agar medium, so we have bactopeptone, protease peptone, that is peptone, we have lactose, bile salts, sodium chloride, neutral rate indicator, and agar agar powder has been added. So the lactose monohydrate, it is acting as a fermentable source of carbohydrate. Okay, so this is about the McConkey's agar medium. And the preparation or the procedure for McConkey's agar media preparation is similar to that of other kind of media. So weighing all the ingredients, putting them in the conical flask except agar, then checking the pH, and then add neutral rate agar, and then add agar, and then put it for sterilization process. So this is about McConkey's agar medium. So let us just examine here the images. You can see here on the right side. So this one is a lactose fermenting colonies. Pink color colonies are formed. This is on nutrient agar media. So this is on nutrient agar media. And this one is a non-lactose fermenting colonies. As we have discussed that white color colonies are formed when incubated on McConkey's agar media, because they are not able to ferment the lactose which is present in the media. So pink color colonies on McConkey agar lactose fermenters. This one is an example for differential media, that is McConkey's agar media. We also have here E. coli, which forms pink color colonies because it is also gram negative bacteria, and Enterobacter aerogenes, Staphylococcus, Salmonella, and Proteus vulgaris also can be grown on this media. So this is how it looks like when bacteria is growing on McConkey's agar media. Next, coming to enriched medium or blood agar medium or blood broth. So what do you mean by enriched media? So enriched media is generally used for cultivation of pasturous organisms which fail to grow on nutrient agar media. That means nutrient agar media is a basic media or basal media which does not contain any special nutritional nutrients. So pasturous organisms, what do you mean by pasturous organisms? Those which require special kind of nutrients for their growth. So it is not able to, that means the one fastidious organisms are not able to grow on nutrient agar media which is a simple or basic media. Then what is the enriched media? So an enriched media is media which contain nutrients are required to support the growth of a variety of organisms, that is variety of organisms including fastidious organisms. And not only that, it is able to allow the growth of wide variety of microorganisms, and generally it is a solid media, because we also have another kind of media, that is enrichment media. Enriched media is a solid medium, whereas enrichment media is a liquid medium. So we do not have enrichment medium, and that is why we are not going to discuss about enrichment medium here. So here, what is the speciality of this enriched media? So enriched means we are adding some special nutrients. Some special nutrients are being incorporated into enriched media. Like for example, blood, serum, egg yolk are being added, and that is how we are enriching the medium for the to support the growth of fastidious organisms, and it will facilitate the growth of fastidious organisms. And some of the examples are chocolate agar, Lofted serum agar, and blood agar. So here we have blood agar medium. You can also see the image here on the right side, that is an uninoculated blood agar medium. So just imagine if you are adding two nutrient agar, if you add blood, then it becomes a blood agar medium, which is red in color, whereas in the previous image, if you have observed, that is McConkey's agar, it is maroon color, whereas here blood agar is usually red in color. So this is the image of an uninoculated blood agar medium, which is placed in the petri dish. So how do we prepare this blood agar medium? So we have nutrient agar. So to this we are adding defibrinated blood. So usually we use sheep blood or goat blood can be used. We have to defibrinate it. So what do you mean by defibrinated? So defibrinated blood is generally added to nutrient agar media, that is for the isolation of some microorganisms here, that is for fastidious organisms. Defibrinated blood of sheep, horse, pig or goat blood can be used for isolation of generally Staphylococcus or Streptococcus species. It has to be defibrinated. That means defibrination means, so in order to the one which is been removed, the blood which does not contain fibrin, that is where fibrin is being removed. So what happens is anticoagulant. So this, if fibrin is present, what does it do? It will help in the clot formation. Okay, so for that purpose what we are doing is we are removing the fibrin, that is what is called as defibrinated blood, and then we are adding the defibrinated blood to the nutrient agar medium, and nicely you can see the formation of hemolysis. Now what is the concept of this hemolysis? Here we are seeing that it helps to differentiate hemolytic and non-hemolytic species, especially Streptococcus species. So what do you mean by hemolytic? Because here certain organisms, especially human pathogens, some of the examples, say for example Streptococcus species, they are able to produce certain toxins. So one of the toxin is hemolysin. HAEMOLYSIN. So hemolysin is the one which will lyse the RBC cells by releasing the hemoglobin. So they are acting on the RBC cells, that is they are lysing the RBC cells. That is how they are able to differentiate. Not all organisms are able to hemolyze, which are growing on blood agar medium. So some are able to hemolyze, some will partially break or lyse the RBC cells, and some does not lies the RBC cells. So it is also acting as a it is helping it to differentiate between hemolytic and non-hemolytic species. One of the examples is Streptococcus species. Now here coming to the procedure. So we know the preparation of nutrient agar media. We can prepare nutrient agar media, and then we are adding 5% of defibrinated blood to the nutrient medium after autoclaving. So you not autoclave it, that means you should not heat it or boil it, then it will become chocolate agar medium, and all the properties present in the defibrinated blood will be lost and will not be taken up by the microorganism which has to be grown on blood agar media. So this is how after autoclaving, and when it comes to around 45 degrees Celsius, the media blood is being added, mix it well properly, and then it is been poured into the petri dishes. So this is the composition of the blood agar media, which is peptone, NaCl, beef extract, agar agar, and then finally after autoclaving, that is sterilization, we are adding defibrinated sheep, horse, pig blood can be added. So just look at this image. So here Klebsiella, Enterococcus, Staphylococcus and Streptococcus are some of the examples which are able to grow on blood agar media, and the type of hemolysis shown by different bacteria here you can see. So here you can see three types of hemolysis patterns which are observed on blood agar media by different bacteria. So here the first one, this is the beta hemolysis, that is beta hemolysis is complete lysis. They are completely lysing or breaking the RBC cells by releasing the hemoglobin. So that is why you can see, so here it is a clear picture of beta hemolysis. So this is a bacterial colony, and this is because of lysis you can see there is not a dark color. So here at this area you can see this is a blood that is which is not being utilized by the bacteria. So here this one is a not used by bacteria, this red color one which is indicating, and here this is a colony. So here this is a colony, these are the colonies here, and around this you can see complete lysis. Lysis of what? RBC cells. So that is what you can see, some zone or clear zones around the colony. That means this bacteria has completely lysed the RBC cells. That is beta type of hemolysis, this pattern. Next, coming to alpha type. So here you can see here alpha type. So you can see the difference here in this picture and this picture. So this is the alpha hemolysis, incomplete. The lysis is not as clear as beta hemolysis. Incomplete hemolysis is been shown, and one of the examples is Streptococcus pneumonia will show alpha kind of hemolysis. The RBC cells are not completely lysed, incomplete lysis occurs. And next, coming to gamma hemolysis. So gamma hemolysis, or this is one of the examples is Enterococcus. This is not at all lysed. That means the bacteria is not going to lyse the RBC cells because it does not contain any exotoxins which has to be released by that particular bacteria, Enterococcus faecalis or Klebsiella pneumonia. They do not produce any exotoxins which can be released into the blood agar medium so that they can lyse the RBC cells. So this is about different species of bacteria which are growing on blood agar medium. Next, coming to algal medium. So we have two algal medium, that is Bristol's medium. The first one is Bristol's medium and the next one is Princeton's medium. So generally this is used to cultivate photoautotrophs, that is algae. So coming to the components. So these are general components, that is potassium dihydrogen phosphate, sodium nitrate, magnesium sulfate, calcium chloride, sodium chloride, ferric chloride, and next one is important one is water, that is distilled water. So all these ingredients are added according to our requirement, and this basically contains, that is these are nothing but salts. So whatever we have seen there, the components, these are all salts, and the phototroph generally they use those salts and just put these media inoculate with the. So what we are doing is, in order to cultivate algae in this medium, Bristol's medium, what you are going to do is you just take a sample, either algae or any pond water where you can find some algae, just take pinch of algae and put it into this flask. So you can see here. So this is the one which algae has not been grown. So after certain period of incubation, you can see these algae, green color algae growing in the medium, Bristol's medium, algal growth in the Bristol's medium. So initially, the medium is watery type. So because all those are salts, we are not going to add any peptone, beef extract or agar is not added. Here we do not use any agar medium because all this is a photoautotrophs. But one important thing is they require sunlight for energy. So what we are doing is we are weighing all the components and pouring in the conical flask, adding distilled water, mixing it properly, adjusting the pH, and then sterilize it, and then let it come to room temperature and add our source of algae. That is, it may be any pond sample or even on soil or anywhere where there is stagnant water for a long time. You can see even on the walls where there is running water, you can see some algae growing on. You can just take that and put in this what do you say, conical flask containing Bristol's medium, and give it some time, generally 30 to 60 days or so, depending upon the sunlight, presence of sunlight, and the growth time which this algae is taking. Okay, so just put it near the sunlight because it requires sunlight. You're not going to place them in inside the room or in the incubator. These are photoautotrophs and they require light for energy. They require energy for their growth. So what we are trying to do is we place near the window where there is provision for sunlight, or outside near where there is provision for sunlight also you can place this. So this is about the algal medium. Generally it takes 30 days, so depending upon the availability of the sunlight. Next one is Springstein's medium. So it is also used for cultivation of algae, that is photoautotrophs. So this also, which is similar to Bristol's medium, mainly consists of salts. Um, so here we are not adding carbon and nitrogen because they are manufactured by the algae itself, and we are not adding them into the medium. Why? Because they are photoautotrophs. They can prepare their food by their own provided sunlight for energy. So next, and the pH which is required for algae to generally grow is 7.2 like bacteria. So this is the composition of Princeton's medium, that is potassium nitrate, magnesium sulfate, diammonium hydrogen phosphate, calcium chloride and ferric chloride. So this is the composition of Princeton's medium, weighing all the ingredients and adding distilled water, sterilizing, checking the pH, and then adding the sample. So this is about media preparation. Along with this, I am also going to share the videos how we generally prepare this media, that is weighing, and after particular period of incubation, how generally the bacteria like on blood agar medium, how they are able to grow. The videos also I am going to share in Google Classroom. You all can have a look. Thank you.